Hello, dear viewers. Welcome to Mr.Zoologist YouTube channel. Myself Nasir. We are going to start a new series of advanced topics of some advanced subjects. These are the part of the masters in zoology. So, our first subject is about the cell and advanced cell biology, or you can call it molecular biology. So, cell is a complex structure which is comprised of subcellular structures. We are going through all these subcellular structures one by one in detail. You might be familiar with the topics, but not in as much detail as we are going to explore right now. So, our first lecture of the advanced cell biology is about mitochondria. Mitochondria are double membrane organelles found in eukaryotic cells. What is meant by they are found in eukaryotic cells but not in prokaryotic cells? The reason is that actually mitochondria evolved in the eukaryotes. That was not the part of the prokaryotic cells. They also do respiration, but they have a different mechanism than those of the eukaryotic cells. In prokaryotic cells, they contain ETC and other protein channels embedded inside their cell membrane. They do not have specific mitochondria organelle in their cell structure. So they are mitochondria in the eukaryotic cells are referred to as powerhouse of the cell. You might be familiar that what is a powerhouse, but here powerhouse mitochondria produces ATP or energy through the oxidative phosphorylation. That oxidative phosphorylation will be discussed in the detailed lecture we are going through right now. So actually ATP is the only form of the energy that is being used by the cells, especially eukaryotic cells, although prokaryotic also produces ATP through the cellular respiration. ATP is the only energy currency of the cell. Cell cannot utilize any other form of energy. That's why ATP is referred as energy currency of the cell. Now we are going to look upon the structure of the mitochondria. That is a double membrane organelle. We have talked it about earlier. It contains an outer membrane and an inner membrane, and inside the inner membrane is a matrix that also contains the F1 particle, that also contains certain enzymes and various components that are involved in different processes. So, double membranous organelle is the mitochondrion. Its outer membrane is permeable, while its inner membrane is very impermeable, means to say it is too much selective as compared to the outer membrane. TCA occurs in the matrix that is inside the inner membrane, and electron transport chain or ETC occurs in the inner membrane. If we talk about the outer membrane, its lipid composition is somewhat about 40 to 50% by weight. It means to say all the lipid contents in the outer membrane varies from 40 to 50%, and it is rich in phospholipids. Phospholipids are also the types of the lipids. These phospholipids that are too much present in the outer membrane are phosphatidylcholine and phosphatidylethanolamine being the most abundant and most rated present. Next is the cholesterol is also present, though less than in the plasma membrane, giving the outer mitochondrial membrane (OMM) a relatively more fluid structure. You might be very well familiar with the fluid that is more viscous than a normal liquid. So these were the lipid components. Next, we talk about the protein composition of the outer mitochondrial membrane. That is about 50 to 60% by weight. It contains porins that form large channels, allowing the free diffusion of ions, nutrients, and small molecules that can be of the up to the size of the 5 kilodaltons. A kilodalton or dalton is the unit used to measure the size of the organelles or the cell. Enzymes involved in lipid synthesis and the conversion of fatty acids into acyl coenzyme A are located in the outer mitochondrial membrane. Outer mitochondrial membrane plays a role in mitochondrial dynamics. such as fusion processes and fission processes, and also involved in the proteins like mitofusion. And after the next to the outer membrane is the intermembrane space. This is a narrow space between the outer and inner membranes. It contains various enzymes and molecules that participate in the oxidative phosphorylation process. It is the gap present between the outer mitochondrial membrane and the inner mitochondrial membrane. It also contains various enzymes that are useful in the oxidative phosphorylation. And after intermembrane space is the inner mitochondrial membrane that contains a lipid about 20 to 25%, and it has high concentration of cardiolipin that is a unique phospholipid and is critical for maintaining membrane integrity and the function of mitochondrial enzymes. It means to say that phospholipid is an associated molecule and that is acts as a carrier molecule that acts as a building block of the inner mitochondrial membrane. Although it also contains phosphatidylcholine and phosphatidylethanolamine, these were present in the outer mitochondrial membrane but are also present in the inner mitochondrial membrane. Their concentration is higher in the outer mitochondrial membrane, but they are also present in the inner mitochondrial membrane, but their amount is so much less as compared to the OMM or outer mitochondrial membrane. Next is the protein composition. Inner mitochondrial membrane is highly protein rich, constituting about 75 to 80% proteins by weight. It houses electron transport chain proteins and ATP synthase, which are involved in the oxidative phosphorylation and ATP production. Transport proteins are also present, such as ADP translocase, which transport ADP and ATP into and out of the cell. And it also contains specific metabolite carriers which carry different metabolites such as pyruvate, citrate, and amino acids. These are carried by carriers. These are actually the metabolites. They are carried by specific carriers which are the proteins in nature. Inner membrane is highly convoluted and forms numerous folds. These folds are called cristae. Cristae significantly increase the surface area of the inner membrane, which is essential for the production of ATP. The inner membrane is impermeable to most ions, and it means it does not allow the free movement of ions and molecules, and it contains the proteins involved in the electron transport chain or ETC. Cristae are actually the inner mitochondrial membrane folds. They are the sites of the electron transport chain where the transfer electrons through a series of protein complexes generates a proton gradient. That proton gradient drives the ATP synthesis. We are going to look upon the ATP synthesis through this proton gradient in the next coming slides in detail. F1 particles are studied, splendid or stranded inside the surface of the cristae. They are the part of ATP synthase complex, which is an essential enzyme complex found in mitochondria. Actually the ATP synthase complex plays a crucial role in the production of ATP or adenosine triphosphate. That is the primary energy currency of the cells. Next is the matrix is the central compartment enclosed by the inner membrane. It contains a variety of enzymes, DNA, ribosomes, and other molecules needed for the production of ATP as well as the citric acid cycle, which is also known as the TCA or known as the Krebs cycle and fatty acid oxidation. And if we talk about the DNA here, that DNA is not the DNA of the cell. That is the DNA of the mitochondria. As you are very well familiar that DNA is a self-replicating organelle. It contains its own DNA. It can replicate on its own. Mitochondrial DNA have their own DNA, which is a circular double-stranded molecule. It contains genes encoding essential proteins involved in oxidative phosphorylation. Mitochondrial DNA is inherited exclusively from the mother. That does not come directly from the father. It is actually the part of the inheritance from the mother. Protein encoding genes. Human mitochondrial DNA encodes for 13 essential protein coding genes that are involved in the ATC and oxidative phosphorylation, which are crucial for the production of ATP, the cell's primary energy source. These genes encode proteins such as like cytochrome C oxidase, NADH dehydrogenase, and cytochrome B. These are the part of the ETC. And ribosomal RNA genes contains 2 ribosomal RNA genes that are involved in the structure and function of the mitochondrial ribosomes, whereas 22 transfer RNA Genes encode for proteins that are responsible for transferring amino acids to the growing protein chain during protein synthesis. So mitochondria have their own ribosomes which resemble bacterial chromosomes and are responsible for synthesizing some of the proteins needed for mitochondrial function. Mitochondria contain a vast array of proteins, many of which are imported from the cytoplasm. It means to say, although mitochondria produces its own proteins, but not all the proteins of the mitochondria is produced by itself. Many of large bulk amount of the proteins are exported from the cytoplasm to the mitochondria matrix, including the electron transport chain, ATP synthesis, and transport of molecules in and out of the mitochondria. So that you can see in the diagram that almost more than 600 mitochondrial proteins are exported from cytoplasm into the mitochondrial surface. So now we are going to talk about the evolutionary status of mitochondria, or you can also say that how eukaryotic cells developed first mitochondria in their body or in the part of their system or in their cytoplasm. That is explained by the endosymbiotic theory. According to the endosymbiotic theory, mitochondria are believed to have originated about 1.5 billion years ago when an ancestral eukaryotic cell engulfed an aerobic free-living alpha proteobacterium. That was a bacteria. This engulfed bacterium established a symbiotic relationship with the host cell. That host cell was eukaryotic. Over time, this symbiotic relationship evolved within the host cell, providing protection and nutrients to the engulfed bacterium, while the bacterium generated energy through oxidative respiration. So, that was the association in which the bacterium became the part of the eukaryotic cell system. So, as this symbiotic relationship evolved, the engulfed bacterium gradually lost many of its prokaryotic characteristics because that was a bacteria. Alpha proteobacterium that was prokaryotic in nature, so it became to lose its characteristics, bacterial characteristics, and became an integral component of the host cell. This included the loss of many genes. It means that bacteria lost many of its genes as the host cell provided an environment with less selective pressure to maintain a complete genome. That bacteria was containing its own DNA when it became the part of this eukaryotic cell that first engulfed it, and that engulfed eukaryote did not disrupted its DNA, but many of its genes were turned off, which were responsible for its prokaryotic behavior. So this endosymbiotic association of the mitochondria eukaryotic cells developed more successfully. So the eukaryotic cells that successfully formed this symbiotic relationship with ancestral mitochondria gained a significant advantage, means that they become more important as compared to the previous eukaryotic cells. They had a more efficient and reliable energy production system which enabled them to thrive in diverse environments, which means to say with the evolution of the mitochondria inside their body, inside within their cytoplasm, they were able to respire aerobically as well as anaerobically. So, several lines of evidence support the endosymbiotic theory. These include the similarity between mitochondrial DNA and the DNA of modern alpha proteobacteria, the similarity in the structure and function of mitochondria and bacteria, and the fact that mitochondria divide independently of the host cell. So, the establishment of this endosymbiotic relationship marked a significant turning point in the evolution of life. It enabled eukaryotic cells to harness or produce the energy of oxidative respiration, leading to increased complexity and diversity in the eukaryotic lineage. It means to say the eukaryotic lineage gained more success after this symbiotic association, endosymbiotic association development. That was the turning point in the evolution of the complex eukaryotes. So mitochondria are now integral to eukaryotic cells, serving as a powerhouse responsible for producing the majority of cellular energy in the form of adenosine triphosphate. So high energy electrons are generated via the citric acid cycle or the Krebs cycle, as you can see in the diagram that acetyl CoA or citrate is formed first when acetyl coenzyme A combines with the carbon dioxide. So that forms citrate, a water molecule is released, and in the third step, in the second step, citrate is transferred into the cis isocyanate, that is the isomer, and that also transfers into another molecule that is also the isomer of citrate, that is isocitrate. And next, isocitrate is oxidated into the alpha ketoglutarate, which is further reduced into the succinyl CoA. Succinyl CoA is reduced into the succinate that produces and the intermediate GTP. That GTP is actually the output of the Krebs cycle. And succinate is further reduced into the fumarate. Fumarate is converted into the malate, and malate into the oxaloacetate. That was the precursor of the Krebs cycle. Actually, acetyl coenzyme combines with the oxaloacetate, and that cycle keeps on repeating. Each Krebs cycle produces GTP. One GTP is equivalent to one molecule of glucose. So now we are going to discuss about the remaining topics. What are the electron carriers? There are 5 types of electron carriers which are involved in the electron transport chain. Actually, ETC is the major part of the energy production or the cellular respiration. So these are the electron carriers which are present inside the mitochondria. We are actually discussing the mitochondria as a powerhouse of the cell that produces ATP. So our main focus is the production of the ATP. That ATP is being carrier mediated through various series of steps, such as you have seen the Krebs cycle. That Krebs cycle actually the part of the mitochondria. So there are 5 types of electron carriers which are involved in the electron transport chain, such as flavoproteins, cytochromes, 3 copper atoms, ubiquinone, and iron sulfur atom. If we talk about the flavoproteins, these are consist of polypeptide bound tightly to the one of the related prosthetic group, either flavin adenine dinucleotide or flavin mononucleotide, FAD or FMN. These have the ability to accept and donate 2 electrons and 2 protons simultaneously. That is the structure of an FAD and FADH2. On the left side, you have an FAD molecule, and if you add 2 proton and 2 electron, that will be oxidized, and that will form an FADH2 molecule. As you can see in the diagram, that nitrogen double bond splits, and it adds 1 hydrogen atom, and as well this addition of hydrogen is known as reduction, whereas addition of hydrogen and addition of electron is actually the part of the reduction. That will be the reduced form, and if you reverse it in the left, from right to left, that will be the oxidized form. And next is the cytochromes. These are proteins consist of heme prosthetic group. The iron atom undergoes reversible transition as a result of acceptance and loss of single electron. As in the previous we have discussed, they accept and donate 2 electrons, whereas that cytochromes accept and donate single electron or proton. There are 3 types of cytochrome: cytochrome A, cytochrome B, and cytochrome C. These all are different from each other by substitution within the heme group. That means to say their heme group is different. As you can see in the diagram, only 1 electron or 1 hydrogen atom is hydrogen ion is being donated inside or outside, means to say being oxidized or reduced by single electron or single hydrogen ion. Next are 3 copper atoms. These are all located within a single protein complex of the inner mitochondrial membrane. They accept and donate a single electron as in the alternation of copper atoms. Means to say that 1 copper atom is replaced by only 1 hydrogen ion or 1 electron. Or you can also say that the hydrogen ion is actually a proton. So ubiquinone is a lipid-soluble molecule containing a long hydrophobic chain composed of 5 carbon isoprenoid units. Like the flavoproteins, these have the ability to accept and donate 2 protons or 2 electrons simultaneously. The partially reduced molecule is called ubisemiquinone, whereas the fully reduced, which contains two added proton or electron, is called ubiquinol. As you can see in the first diagram, that is the ubiquinone. In the second diagram, you are looking at the structure that has added one electron or one proton, that becomes UB semiquinone partially reduced. And if we add another one proton or electron, that will be become fully reduced, that is the ubiquinol. So next we are going to discuss about is the iron sulfur atom or protein. This is an iron containing molecule in which iron is not present within the heme group. It is linked to inorganic sulfides ions as a part of an iron sulfur atom. The complex is capable of accepting and donating of a single electron. That is the structure. As you can see, the corner on the front side contains iron, and on the left side is of sulfur, and that iron actually contains the sulfur atom. That is why it is an iron sulfur containing a molecule. And now we are going to look upon the importance of reduced coenzymes in the formation of ATP, and that process is called as chemiosmosis. First of all, high energy electrons are passed from FADH2 or NADH to the series of electron carriers in the electron transport chain. The controlled movement of protons back across the membrane through an ATP synthesizing enzyme provides the energy required to form ATP from ADP. Next, second is electron transport. Five of the 9 reactions of Krebs cycle in the matrix are catalyzed by dehydrogenases that transfer pairs of electrons from substrates to coenzymes, such as NADH or FADH, that transfers electrons to ETC or electron transport chain. So NADH and FADH2 dehydrogenases are located in the inner membrane of mitochondria. Electron transport complexes are 1, 2, 3, and 4. They are fixed in place 1, 2, and 4, in which the transfer electron is accompanied by major release of free energy. Ubiquinone lipid is lipid soluble. Cytochrome C is soluble protein in the intermembrane space that moves within or along the membrane. And now we are going to discuss about the electron transport chain of the inner mitochondrial membrane in detail. The respiratory chain consists of 4 complexes of electron carriers and 2 other carriers. We have discussed the 4 carriers: cytochrome complex 1, 2, 3, and 4, and other carriers are ubiquinone and cytochrome C that are independently disposed, spread inside the plasma membrane of the mitochondria. Electrons enter the chain from either NADH. If they come from NADH, they will be the part of the complex 1 first, or if they come through the FADH2, they will be the part of the complex 2. Electrons are passed from either complex 1 or 2 to the ubiquinone, which exists as a pool within the lipid bilayer, separated inside the lipid bilayer. Electrons are subsequently passed from reduced ubiquinone to complex 3 and then to the peripheral protein cytochrome C, which is thought to be mobile, which means that is in the continuous movement molecule. Electrons are transferred from cytochrome C to complex 4, that is also known as cytochrome oxidase, and then to oxygen, that is actually the final acceptor of the electrons and forms water molecule. The sites of proton translocation from the matrix to the cytosol are indicated. The proton numbers shown are those generated by each pair of electrons transported, which is sufficient to reduce only one half of the O2 molecule. Half O2 molecule is actually produced from the water molecule. The Q cycle can be divided into 2 steps, each of which leads to the release of 2 protons into the cytosol. That is actually the diagrammatic chemiosmosis which we were discussing in the previous slide. That is the first cytochrome complex 1, and the next is the cytochrome complex 3, and that does not come the cytochrome complex 2, that comes after the cytochrome complex 2, after the 3, and after the 2, that is the cytochrome complex 4. And with, as you can see in the diagram, with each 2 hydrogen ions or with each 2 electrons transferred, there is the production of 4 protons, and 4 protons are produced in the first cytochrome complex, and they are released into the outer surface or intermembrane space. And the accumulation of the 4 from the first accumulation of the 4 proton from the cytochrome complex 3 and 2 from the cytochrome complex 4 will accumulate 10 protons in the intermembrane space, and the matrix is now more negatively charged, whereas the intermembrane is more positively charged due to proton accumulation. And here you can see the UQ, that is a ubiquinone. Ubiquinone is transferring electrons, that is the carrier of the electrons that mediates electrons inside the membrane of the mitochondria. And here you can see the counts of the different DNAs like mitochondrial DNA and nuclear DNA is mitochondrial DNA in the complex one is about 7 and nuclear DNA is about 38 and total DNA are 45. And after the total count of the DNA of the mitochondria and the nucleus, below is the molecular mass of the overall complex 1, 2, 3 and 4. And actually the arrangement is of different. First comes the cytochrome complex 1, then 3, then 2 and then 4. And you can here see the complete count of the mitochondrial DNA, nuclear DNA, total DNA and their sizes in detail. And these protons we are going to discuss next, that becomes the cause of the chemiosmosis, that becomes the source of the oxidative phosphorylation that in turn produces ATP. These protons will drive gradient, these protons will traverse inside and that will run the whole ATP synthase complex structure that will in turn produces ATP. That is the complex structure that we are going to discuss next. But that's not as much complex as I am going to teach you in the current video. That is the structure of an ATP synthase complex embedded in the membrane of the mitochondria. We are going to discuss in detail. That is the head portion is known as the F1 particle and tail portion is known as the FO. And we are going to discuss it about in detail. That is the complete structure of the ATP synthase. It contains 3 alpha units and 3 beta units and 1 gamma stalk, as you can see in the yellow structure that is central to the these three units of the alpha, beta and gamma is a stalk and C is the structure we are going to discuss in detail in the upcoming slides. And epsilon is used to attach the gamma stalk to the C subunits, which is the base of the ATP synthase enzyme. And here is the rotor part, is the bottom part, FO part. We are going to discuss it now. No need to worry about. That was the schematic structure of the bacterial ATP synthase. The enzyme consists of 2 major portions called F1 and FO. The F1 head consists of 5 different subunits in the ratio 3 alpha, 3 beta, 1 epsilon, 1 gamma. And the alpha and beta subunits are organized in a circular array to form the spherical head of the particle. The gamma subunit runs through the core center of the ATP synthase from the tip of F1 down to the FO. FO is actually the base and F1 is the upper part from the base. To form a central stalk, the epsilon unit helps to attach the gamma stalk subunit to the FO base. The FO base, which is embedded in the bacterial plasma membrane, consists of 3 different subunits in the apparent ratio 1A, 2B and almost 10 to 14 C subunits. The C subunits form a rotating ring within the membrane. The paired B subunits of the FO base and the subunit of the F1 head form a peripheral stalk that holds the subunits in a fixed position. The A subunit contains the proton channel that allows protons to traverse the membrane. Means to say that what traveled into the intermembrane space will now enter into the matrix of the mitochondria. The mammalian enzyme contains 7 to 9 additional subunits of whose functions are not well established right now. So that is the diagram you get in through the microscope at the first size. So you can see in the top structure that is the contains the F1 particle that contains 3 alpha units and 3 beta subunits, whereas gamma stalk is central to them and epsilon is used to attach that to the base, and that base is a rotating C ring unit, contains C units and that are supported by A subunits. So basis of ATP formation according to binding change mechanism. We are going to discuss in detail the chemiosmosis through the binding change mechanism. So, binding change mechanism is a key concept in understanding how ATP synthase, the enzyme responsible for producing ATP or adenosine triphosphate, synthesizes ATP during cellular respiration. ATP synthase is located in the inner mitochondrial membrane in eukaryotes or the plasma membrane in prokaryotes. I have told you earlier in the start of the lecture. In prokaryotes, there is no mitochondria. They contain their ETC or chemiosmotic protein channels in their membrane and operates using a rotary mechanism to catalyze the formation of ATP from ADP. ADP is actually adenosine diphosphate, which becomes ATP when one inorganic phosphate is added into the ADP. So this process is driven by a proton gradient that often referred to as the proton motive force across the membrane. So that was the FO domain. We have discussed it earlier, and the top and at the bottom is the FO domain. The binding change mechanism was proposed by Paul Boyer, and it explains how the F1 catalytic domain of ATP synthase produces ATP through conformational changes in its subunits. So ATP synthase consists of two major domains. F1 domain, that is also known as catalytic domain, that is the most active part in the ATP synthesis. And FO domain is a rotary motor that is embedded in the inner membrane and acts as a proton channel that allows the protons to traverse from intermembrane space into the matrix of the mitochondria, facilitating proton flow across the membrane. The F1 domain has three catalytic beta subunits which interact with three non-catalytic alpha subunits. It means to say actual structures producing ATP are the beta subunits. Alpha subunits are just to maintain their structure. Each subunit undergoes conformational changes during the binding change mechanism. FO domain is composed of a rotating C ring and a stator. That stator is made up of A subunits. And actually A and B subunits are not the rotating, whereas the C are the rotating subunits, which allows the proton to traverse from intermembrane space into the matrix of the mitochondria. The rotation of the C ring is driven by protons moving down their electrochemical gradient, I have told you as earlier. Next, the binding change mechanism explains the coupling of the rotational motion of ATP synthase with ATP production, which means to say with the motion, with the rotation of the C ring, there is a conformational change in the beta subunits of the F1 domain, that was the catalytic domain. So we are going to discuss it into the three conformational states changes in the beta subunits. First of all is the loose state, L state, that the beta subunits bind ADP and inorganic phosphate very weakly. Next is the tight state. In this state, beta subunit binds ADP and inorganic phosphate tightly, facilitating the actual formation of ATP. And third state is the open state, O state. The beta subunit has a very low affinity for ATP, causing the newly formed ATP to be released. So the steps in ATP synthesis: proton movement through the FO domain. Protons flow through the FO domain via the C ring down their electrochemical gradient from the intermembrane space where there is high concentration of the protons to the matrix where there is low concentration of the protons. So this proton flow causes the C ring to rotate, which means to say, as we have seen in the previous slides, there are about 10 to 14 C subunits. So these protons traverse from each subunit, and these subunits drive the central gamma stalk that were embedded in the center or core of the beta and alpha subunits. So rotation of the gamma subunit. The rotation of the gamma subunit is central to the driving conformational changes in the beta subunits of the F1 domain. As I have told you earlier, that if this gamma stalk rotates, the alpha and beta subunits will change their places. Their conformation will change. Their conformation will change from all three stages: LTO, loose state, tight state, and open state. We are going to discuss them one by one. So in the loose state, ADP inorganic phosphate bind weakly to the active site of a beta subunit. The other beta subunits are in different states. So only one beta subunit can be in the L state at a time, or loose state at a one time. As the gamma subunit rotates and induces a conformational change in the beta subunit transitions from the loose state to the tight state. In the tight state, ATP and inorganic phosphate are tightly bound, and this tight binding induces a favorable environment for ATP formation. ADP and inorganic phosphate are catalyzed into ATP without requiring direct energy input. The energy for this step comes from the conformational change itself. So release of ATP occurs in the open state. After ATP is formed in the tight state, further rotation of the gamma subunit causes the beta subunits to change into the open state. In the open state, the affinity for ATP is very low, causing the newly synthesized ATP molecule to be released into the mitochondrial matrix. So the cycle repeats, gamma subunit continues to rotate, and the three beta subunits cycle through the loose, tight, and open states in sequence one by one, allowing ATP synthesis to continue as long as protons are flowing through the FO domain from intermembrane space into the matrix of the mitochondria. Each complete 360-degree rotation of the gamma subunit typically produces 3 ATP molecules, one from each beta subunit. So the searing rotates in increments as protons bind to pass through the FO domain. This rotation is coupled to the movement of the gamma subunit in the F1 domain, which induces conformational changes in the beta subunits where ATP synthesis occurs. For every full rotation of the searing, the gamma subunit also makes a full rotation. This rotation drives the sequential transition of the three beta subunits through the loose, tight, and open state, producing 3 molecules of ATP per full rotation. So, since the full rotation of the searing is 360 degrees, the angle of rotation for each proton can be calculated based on the number of C subunits. For example, if there are 10 C subunits in the searing, each proton passing through would rotate the searing by 36 degrees. If we divide 360 by 10 C subunits, that will be 36. And if there are 8 C subunits and we divide 360 over 8, that will be the 45 degree. So that was the structure we have discussed in detail. The F1 particle, the FO domain. F1 particle was the catalytic site. FO domain was the rotary mechanism or rotary site. So that was all about the binding change mechanism and about the structure of the mitochondria and its cellular respiration, or the key mechanism through which it produces ATP. So we will meet you in the next lecture with a detailed next structure or organelle of the cell. Thank you very much for attention.